Okay. I guess yeah, you can
Okay.
take.
Okay, I can get started then. Hi, everyone. Let's get started. So I appreciate you all uh, for taking the time here to attend my presentation today. I'm Today I'll be talking about a fascinating software called AlphaFold, which is for protein structure prediction. So this is a cutting edge technology that has potential to revolutionize our understanding of how the protein structures are predicted and finally to know their functions. And I hope you find this topic as engaging as I do. And I look forward to lively discussion. So uh, in the outline, I'll first talk about what is the protein structure problem and how does AlphaFold work and some of the results and challenges of AlphaFold 2. Uh, to define the problem, let me take you back to 1972 when Dr. Christian Anfinsen made a bold statement in his Nobel Prize acceptance speech. He proposed that a protein's 3D structure should, in theory, be determined solely by its one-dimensional amino acid sequence. Let me get the pointer option. Uh, okay. Solely by its amino acid sequence. So this hypothesis led the Fuse on a 50-year journey uh, to achieve an algorithm which can predict a protein's three-dimensional structure. But uh, there are various challenges in realizing his vision. Uh, so one major obstacle is the sheer number of ways a protein can fold before reaching its final structure. That is the Leventhal paradox. Leventhal calculated that the number of ways a protein can fold before it takes up one of the conformation with lowest energy is around 10 per 300, I believe. But we all know that proteins fold spontaneously and sometimes even within the micro to milliseconds. So the contradiction is, let alone by the brute force, it can't go through the 10 per 300 conformation. So there are ways nature has figured out a protein takes up a particular form. So this contradiction points to the questions: what determines the structure of the protein based on the amino acid sequence? And why do proteins fold so quickly? And how can a computer algorithm predict protein structure from their sequence? Uh, so, and today, as I mentioned, I'll be talking about AlphaFold. You might have all heard of AlphaFold, which is from DeepMind. With AlphaFold entered a CASP. CASP is Critical Assessment for Techniques of Protein Structure Prediction, which conduct seminar type of things every 2 years once. And we can see that on the left, if we compare the global distance test versus number of years CASP has um, taken place, we can see that in 2018 and 2020, both in 2018, AlphaFold 1 and 2020, AlphaFold 2 has won the competition. So how did AlphaFold make this breakthrough? Uh, so before going into the AlphaFold's integration and how it works, I want to give like a brief overview of how we can use the existing data to produce 3D structures. So I don't know if you have heard of multiple sequence alignment. I'll just give an example here. Um, so the typical process in multiple sequence alignment involves starting with a specific sequence. For example, let's consider this is the human sequence on the top row, and then we search through a genetic database for similar structures. For example, any other type of animal like fish here or a dog, and multiple sequence alignment is comparison between all of these sequences. So once we have aligned these sequences, let's take a look at this matrix and try to identify some of interesting properties. For example, when we examine just a single column, the column with residue C at position 2, so we see that this position is conserved. Uh, uh, so conserved positions may preserve some characteristics. Maybe the characteristic could be the residue C can be either hydrophobic or hydrophilic. Just let's take it as hydrophilic, for example. So we'll know that if they are hydrophobic, they are likely to be inside the protein. And suppose if this position was hydrophilic, it wants to be outside the protein. So that is one of the information that we can get from looking at this multiple sequence alignment. Another useful information is to look for residues that are in contact, and they often mutate together. So for example, if we look at the conserved positions, this purple block and the blue block here, the columns. So if we consider the relationship between these residues that are in contact, for example, if we consider this purple residue here is positively charged and this one is negatively charged. So they both want to mutate together. And suppose if that doesn't happen, if both become positive, the protein will fall apart because of could fa- could fall apart because of the repulsions between them. So they tend to mutate together. And most probably this distance between the co-evolved positions will be conserved during the evolutionary track. So as a result, we see a lot of correlation information in multiple sequence alignments, which are very useful in predicting the three-dimensional structure of the protein. Um, so yeah. So next, I'll talk something about pairwise representation. So pairwise representation can be thought of as containing information about the relative positions of amino acids in the chain. So, like for example, you can consider this as um, pairwise distances, which is distance measured between the C alpha carbon atoms in the protein chain. So this representation is used to predict distances between amino acids in the chain. Uh, in AlphaFold, they use something called bin distance distribution, where the position of each amino acid residue is binned from 1 angstrom to, I think, 30 angstroms, around that region. So AlphaFold structure incorporates the during the training process, it's a machine learning model. It incorporates physical and geometric understanding of the protein structure. So the residues that are close in folded protein structures, they learn to communicate effectively compared to residues which are not in which are not in contact. When I say contact, through one-dimensional amino acid sequence, we don't know if we don't know which residues are interacting. So the position 1 could be interacting with the end position 2. So the AlphaFold network learns a graph type of thing where it determines which residues should be interacting. So next, next I want to mention something about the templates. This is not very important during the inference mode of AlphaFold. So when I say inference, I am talking about during the structure prediction, not during the training. So during training, AlphaFold used templates from PDB database, which has around 170,000 experimentally resolved protein structures. So templates more or less are not that um, useful or required by AlphaFold during the inference mode. So so it mainly uses multiple sequence alignment during the inference mode to predict the protein structure, um, uh, to produce the final 3D structure. So now let's look at the AlphaFold 2 architecture and what it is. So it has 3 parts, mainly embedding part, which is for the input evaluation. Then there is a trunk part, uh, and then there is a heads part where it is also called a structure module, where uh, there are 2 machine learning models here. One is the trunk and the heads. Um, heads is like for 3D structure prediction, and the trunk is for learning about multiple sequence alignments and understanding how pairwise representation between the residues. So it essentially communicates the MS multiple sequence alignment data and the residue interactions in the pairwise representation. So all right, let's look in detail about the embedding and inputs first. Let's start off with an input sequence. For example, this input sequence with some colored residues here. So this input sequence will be running against genetic databases and and produces multiple sequence alignment. So this search, as I mentioned before, finds evolutionary related sequences and align them and put them into a matrix. So this is the key input for the network. So for a computer scientist, these are all just numbers. Uh, they convert the sequences, which are alphabets, into numbers, and then they produce something called embedding, um, uh, which is a matrix of uh, converted sequences into numbers. So in addition, as I mentioned before, there is something called pairwise representation, which you can think this as distance between the residue positions. So they generate pair information by simply combining the pairs of residues. Um, this you can thought of as like a so position 1 to the position 1 here to position 2. It's like dot multiplication in the matrix. Finally, sorry for that. Finally, this information may also include template structures. Um, so to finish up, we search against protein data bank database to see if there are any good templates. So the next part of this input is to feed the input network, which are matrix of numbers, where it uh, did multiple sequence alignment and pairwise representation between each residue, and put them into a network called Eva Farmer. So this one is they called it Eva Farmer because this is a machine learning model called transformers. They named it as Eva Farmer. So here, the pairwise representation process extracts information about residues in contact with one another from multiple sequence alignments. So there is always communication between MSA representation and the pairwise representation. So after this process, the multiple sequence alignment and pairwise representation will be thoroughly processed, which they use as input in the Eva Farmer block, which has like 48 blocks. And specifically, what happens here is that we take the first row, for which we want to produce the three-dimensional structure of the protein, and along with the pair representation here. So we take this row and the pair representation and feed it into the next step of the process, which is a structure module to produce a final 3D structure. So coming into the sorry about that, structure module here, which is another neural network to predict the position of each residues in a specific parameterization, which I'll elaborate on later in the presentation. So the structure module then predicts the dihedral angles for the side chain, which are angles that describe how side chains are attached to the overall structure. So I want to mention one more point about this architecture. So that is recycling. So recycling involves taking the output of the network and feeding them back into the network again. So once you produce the 3D structure of the protein, you take that coordinates and feed it back to the input. So generally you can set this as a variable while predicting the structure. Generally it is around 3 times. So you get, so essentially we take this final output structure and input it back into the same network and repeat this process. So for example, if the recycling is set to 3 times, this results in a total of 4 executions of the model, producing a 48 into 4, 192 blocks. Essentially, this will increase the accuracy of your final structure. So if the recycling is 3 times, you get around 5 structures, which are ranked 1 to 5, rank 1 being better structure. So as I mentioned, I wanted to give you like how the structure module predicts the backbone, for example. So predicting the backbone of a protein structure, what AlphaFold uses is the a representation called orientation plus XYZ coordinates of C alpha in the backbone. So the bond lengths between C alpha and these nitrogen and carbon atoms here, they are fixed, as well as the bond angles. And the variables here are dihedrals. So to represent a dihedral, you'll need 4 atoms. So it takes up a random orientation and varies this dihedral angle such that it attains the feasible minimum energy configuration to get the side chains, correct side chains. AlphaFold uses amber minimization. And briefly, I want to also mention about some of the outputs we get from AlphaFold. One is per residue confidence index. So the first one here is per residue confidence index, which indicates how confident the network is in its prediction for each residue. So from this blue residue to the next residue, and it tells whether how well the residue is confident compared to number of known structures. So the second output is the predicted aligned error output. This allows us to determine whether two residues are positioned correctly relative to each other in the prediction. So essentially, it tells us how the interactions happen in the multi-chain structures like homotrimer and others. I'll give an example on this in the result section, but these are the two important metrics that AlphaFold 2 produces to know the accuracy of our protein structures. So let's take some of the results of AlphaFold 2. So this prediction is for USP7 protein structure predicted by AlphaFold 2. So AlphaFold 2 has unique feature of not only providing the accurate models of individual folding units, but also indicating the dynamic movement between them. So as I mentioned, this USP7 structure is a great example to illustrate that predicted alignment error. So here the prediction highlights the preferential interaction between N-terminal N-terminal domain called this TRAF, this region here, with the catalytic domain right here. So we can see that this square part here is interacting. It tells us this is a different domain from this one, and how they're arranged in 3D space. It also gives some hints about where are the other domains arranged. In this example, they just briefly mentioned them as 1, 2, 3, 4, 5 for this. So these predictions are supported by structural analysis from X-ray crystallography and NMR spectroscopy as well. Sorry about that. And so I want to show you briefly, we have AlphaFold 2 in our lab, and we have conducted some simulations. I want to show one example on cut4 monomer. If we look at the AlphaFold 2 predictions, we have two representations here. One is predicted, one is the IDDT versus positions value, and you can see that at 0th position, it is not very well predicted. The value is like 20, but the monomer structure in this case has good predictions in this region here. And the red region, it is not very confident. So our goal now is to run all item simulations on this and try to get like better structure. On the top, again here, it is very confident in this region, but between the regions, suppose this blue part corresponds to this uh, positions here, and the badly predicted regions, which are these parts, it is not very well confident about it. And the results become hard when we put in like a tetramer structure, for example. This structure is a tetramer structure of the cut4 protein, and uh, we can see that it. I've also put model confidence index here, and we can see that it is not very confident. So even the PLDT show from 0 to this one has a sequence of length 1008. So from here to here corresponds to one monomer unit. So it drops pretty significantly. And even the predicted alignment error is not very well. So one way to improve the accuracy is to use like a large database or increase the number of recycles. So that's what we are trying to do now to get this structure more accurate. And coming into challenges, AlphaFold 2 does not right now has capability to predict metal ions, cofactors, or other ligands. And post-translational modifications like glycosylation are not predicted currently using AlphaFold. And AlphaFold can't take considerations of pH, temperature fluctuations into considerations. And right now, DNA, RNA, and their complexes are not predicted under AlphaFold. So the current multimers are trained only for multimers of length 3000 residues. So if we compare that to our residues here, which has like more than 4000 residues, the accuracy drops significantly. Yeah, that's about it. And thank you. And if you have any questions.
So, Anu, I have a question.
Yes.
It was a nice talk. I got so much idea about AlphaFold and how it works and what is the architecture and so on. But I know, apart from the protein structures that are already present in the database of AlphaFold, will you be able to like add your or like make your own structure, and with the help of AlphaFold, will you be able to make a prediction, like confidence curve, or else the error?
So for example, as I mentioned, this is one of the structures which is a tetramer, and the structure of this is not experimentally known.
Okay.
Right? So this has to be so as we can see, the confidence is pretty bad here. But one way to increase the accuracy of this model is to increase the to include all the databases and to increase this recycling. So keep feeding it back into the loop until it produces like very little error. And but the scaling with AlphaFold 2 is not that great with the CPUs, right? But yeah, so essentially this is not the correct structure. It is not always producing the right structure. Especially with the multimers, it has only been trained for multimers with around 3,000 residues. This itself has over 4,000 residues. So the accuracy drops. But if we look at the monomer, which has also not been non-experimentally resolved, but it is very confident about this.
Okay. And when you say that it is not considering the temperature, pressure, so how do you think if your structure may be affected by temperature and pressure, but the AlphaFold is not taking those into consideration? So how
Yeah.
think about like how much accurate structure you will be getting without considering the physical factor condition?
Yeah, so I will have to look into details about if there is any paper on the effect of pH and temperature, but it predicts fairly accurately for a smaller monomers, smaller proteins, and this is entirely based mostly on multiple sequence alignment in the inference mode. However, during training, it takes into all the known structures in the PDB. But I am not aware of the effect of pH or the temperature. It cannot take that as input, is what I meant.
Yeah.
Like to produce protonation states and other factors, it does not include that. But we can take input. We can take the 3D structure of this protein, for example, and conduct all atom simulations and include temperature or other factors.
Yeah. I'm guessing it's
In
like a temperature where the structures were resolved.
Oh, okay.
Yeah, probably.
Yeah.
Because all the input is like.
It depends probably on the protein. It's either 25 or 37 Celsius.
Yeah.
Oh, okay.
Yeah, that's
Probably
kind of,
around.
it's either body temperature or ambient temperature, depending
Yeah.
on what the protein is doing.
Yeah.
Do you guys have other questions? Actually, I have a few questions. So, going back to one of the results slides with the ranks, I wasn't sure what the rank 1, 2, 3 that meant actually.
This one?
Yeah. What do the different ranks mean?
Oh, ranks. So as I mentioned, it undergoes some 3 recycles, right? But you can change this. So rank 1 corresponds to for example, it may corresponds to structure of first recycling.
Oh, I see.
Right.
Okay.
And rank 2 is the second one, third. So rank 1 generally means the structure that corresponds to minimum error.
I see.
Yeah. So if it's 3, you get 5 structures, right? 1 initially, and then you do it 3 more times. So overall, you will get 5 structures in the end. So that's why it's rank 1 to 5 in this example.
Oh, okay, I see. And then my other question was, is AlphaFold 2 good for disordered proteins?
Disordered proteins. I'll have to look into an example, but I think Professor Faller can talk about. I don't know if you remember Shiaki's work. He used AlphaFold 2 for disordered proteins, right?
The problem is AlphaFold doesn't care, but
Yeah.
the training set in the PDB is strongly biased towards ordered proteins. Therefore means the training set is way stronger on the ordered protein side. So therefore it has an ordering bias. Like essentially any machine learned protein structure prediction has an ordering bias.
Okay, makes sense. Makes sense.
Yeah.
So that's exactly, we have a collaboration with Priya Shah, Shiaki, one of her students.
Oh, yeah.
Is also using AlphaFold together with us. And we have a protein which has a significant disordered part. And there we definitely ran into some issues that it looked more ordered than it should.
I see.
But then if you run MD on it, it actually it helps. So
Okay,
essentially what we normally do, we use AlphaFold to generate initial conditions.
right. That's true. Nice.
Yeah, it's entirely based on the learned based on the, I think, non-resolved, experimentally resolved structures, which are mostly ordered in the
I just have
PDB.
one more question. I don't know the answer to this one, but have you compared AlphaFold 2 with Rosetta? Because I know Rosetta also predicts protein structures.
Yeah, so I haven't done any simulations with Rosetta, but yeah, I know that they also used machine learning similar to AlphaFold to predict structures, and they actually have improved some features on it, is what I read about, but I haven't used it in my research.
Essentially, from what I understand, is that AlphaFold is dominating the machine learning protein prediction at the moment. Won all the last cast competitions easily. And it mainly is because it is the only one which has the complete PDB as training. So it really is trained on all known structures.
Oh, they have the computing power. So
And so it really, there is, as you might know, there is ColabFold is actually a free version where you actually can just put it in a website and run it. It has some limitations on terms of memory, and so we have a AlphaFold version on the cluster running locally. But you can run ColabFold, which is essentially based on the AlphaFold algorithm, but with not the full training set directly off the bat.
Yeah, yeah, and they used this. I don't know if you guys heard of GPT-3 models, the one with the Chat GPT. So they used this model to train AlphaFold, which is very, very good.
oh, okay.
Yeah, it's for unordered data set. So previously they used convolutional neural networks with AlphaFold 1, but yeah, apparently this one gives them very good results in the AlphaFold 2. Yeah, but the problem with is the multi-mesh structures, like it's not very good.
Any other questions? Oh, good. Well, thank you, Anu, for your great presentation. Yeah, we learned a lot.
Thank you.
And if you're interested, we have it. It's installed on, I think, HPC2 now.
It's on HPC2.
Oh,
Yeah.
okay.
So yes, so if anyone needs it, definitely we can arrange that. Because doesn't so it's I think it's installed for everybody by now, or is it now in our partition? I don't know.
So right now, you can reach out to me. I can like tell you how to use it.
Okay.
Yes. Yeah, it's on the HPC2 system under some environment that you'll have to import that in your account.
Okay.
All right. Yeah.
Thank you.
Thank you. Yeah. See you guys next week.
See you. Thank you.
That's why.